Hello everyone. Today we are going in detail for heterospory and seed habit. The word itself gives us idea. Hetero means two different types of spores. They differ in size, structure and function, and obviously they are formed on the same plant. Pterodophytes, they produce only one kind of spores, that is they are homosporous, but in Selaginella, Isoetes, Siletes, Marsilia, Piluraria, then Regnelidium, Salvinia, Azola, Platysma, Heterospory is very common, and that leads to the seed habit which is observed in angiosperms. Then how this heterospory originated in pterodophytes, either due to the reduction of a number of spores within sporangia, or it may be due to the disintegration of some spores in the sporangium. Why this is happen? Because when all the spores are functional, there is competition for nutrition and as well as for the food supply, and few of them they remain small in size. They are the microspores, and remaining spores they get sufficient nutrition for the development, and that's why they are larger in size. They are the megaspores. In Marsilia and the Selaginella, it has been observed that due to the low light intensity, there is a low photosynthesis, and that leads to the low nutrition, and this is one of the limiting factor for the growth of the spore, and they could not grow. This is observed in fossils also, like Lepidocarpan, Calamostachys, Calamocarpan, Stauropteris. They are also there. That means this heterospory is observed in fossils and not originated in only pterodophytes. There are the different evidences for the heterospory, and they are before mitosis as well as the after mitosis. There are different examples for the before mitosis in Selaginella stenophylla, where two megaspore of a tetrad slightly larger than other two. In Selaginella molliceps, one megaspore is larger and remaining three are smaller, whereas in Selaginella erythropus, megasporangium is surprisingly only with one larger megaspore, and there are no traces of aborted spores. In Isoetes, microsporangium is with 1,50,000 to 10 lakh microspores, but megasporangium is only with 50 to 300 megaspores, and these changes or these observations are before mitosis, whereas after mitosis, again in Marsilia, Salvinia and Azola, this heterospory evidences are observed. In Marsilia, the microsporangium is with 64 microspore after reduction are functional, whereas megasporangium, in which out of 64 only one remain functional. Remember, out of 64, 63 they disintegrate and only one remain functional. Same case is observed in Salvinia as well as the Azola. Then what is importance of this heterospory in pterodophytes? That as we know, microspore they gives rise to the male gametophyte, megaspore they gives rise to the female gametophyte, so it is useful for expression of sex at their spore stage. And obviously development of gametophyte is endosporic, and this along with this nutrition is derived from sporophyte. So the development of embryo gametophyte is not affected by external, or we can say ecological factor. Another importance is that megaspore is retained by parent even after fertilization, and this ensures nutrition for the developing embryo. So here embryo can be very well developed and it remain itself into the megaspore. Then this heterospory that leads to the seed habit. As we know, male and female gametes, they fuse to form a zygote that develops into the embryo inside the ovule. Embryo and the rest of gametophytic tissues like integuments and ovary wall, they form a seed, and seed finally germinates into the new plant. In pterodophytes, this evolution of seed habit requires some number of steps. One is formation of two types of spores, megaspore and the microspore. Then reduction in number of functional megaspore to 1 per megasporangium, means out of number of megaspores only one remain functional. Then this megaspore is remain inside the megasporangium until embryo development. Afterwards, elaboration of a apical part of a sporangium to receive microspore or pollen grain for fertilization. And lastly, availability of a sufficient nutrition for the development of embryo. So this is very well observed in the angiosperms. But at the end, the question is that in pteridophyte there are no true seeds, as they are showing the seed habit. Here, the development of female gametophyte, fertilization and embryo development that takes place within the megasporangium. And this is the reason for evolution of seed habit in the pteridophytes, and specifically it is well observed in Selaginella. But it is not to be called as a true seed. Why this is so? Because though megasporangium in megasporangium embryo development takes place, but it is not covered with the integument as in angiosperm. Along with this, there is a no resting stage after embryo development, and this embryo development accompanied with the development of a shoot and the rhizosphere. So this is the reference for the heterospory and the seed habit. With this, we came to know that heterospory, which is with the two different type of a spore, and they differ in the size, shape as well as the function, and this heterospory in pteridophytes leads to the evolution of a seed habit towards the angiosperm, but they are not true seeds because of no resting period as well as the no integuments are there. Thank you.